Hello and welcome everybody to my channel ominibioentrance, and today we will be discussing CSIR PYQs of the year 2022. If you are new to my channel, please do like, share and subscribe. So coming to the year 2022, this exam was conducted in the year 17 to 22. This is shift 1. The basic difference between direct repair and base excision repair is direct repair restores original structure of altered nucleotide without replacement, while in base excision repair, the section of DNA containing the distortion is removed. The correct base is added and resealed. That itself is the basic difference between direct repair and base excision repair. So first one is your answer. Then next one. Which of the following proteins is essential for both the initiation of DNA replication as well as continued advance of the replication fork? It is CDC45 that is involved, whereas ORC, Geminin, CDC6 are involved only in initiation. CDC45 is involved both in initiation as well as elongation. Then next one. In bacteria, many of the tRNA gene do not contain the CCCA sequence found at the 3' end of the tRNA. In this context, which one of the following statements represent the correct explanation? In these tRNAs, amino acylation occurs at the 3' end of the tRNA irrespective of the presence of CCA sequence. That is not correct because CCA sequence is essential at the 3' end, and there must be CCA sequence there at the 3' end, without which amino acid cannot attach. So first statement is not correct. CCA sequence is added to these tRNA transcripts in a DNA template independent manner. That is correct. These tRNAs exploit the process of trans-splicing to include a CCA sequence at their 3' end. That is also not correct because trans-splicing doesn't take place here. The absence of CCA sequence occurred only in the last common ancestor during the course of evolution, and the current day tRNA genes always possesses a sequence to encode the CCA end of the tRNA. That is also not correct. So the correct answer is option 2. Then next one. The following statements are made with reference to DNA replication. Which of the following options represent the correct statement? I forgot to include the options here, but I will give you the correct answer. Camptothecin causes intra and interstrand crosslinks in DNA, leading to stalling of replication fork. Now camptothecin, it is a topoisomerase inhibitor, so it does not cause any intra and interstrand crosslinking. So first option A is not correct. Then next one. Prevention of reinitiation of DNA replication during the same cell cycle is mediated by regulating the loading of the initiator complex ORC. It is not loading of the initiator complex ORT. It is through covalent modification. So B is also not correct. A glutamine alanine mutation in the nucleotide building pocket of DNA polymerase 3 could lead to the incorporation of ribonucleotides in the extending DNA chain. Yes, that is correct. Then, a mutation in the gene encoding topoisomerase 2 could lead to entanglement of DNA daughter strands during replication. That is also correct. Why? Because topoisomerase is involved in removal of supercoiling during replication. So, C and D are the only correct answers here. Then, which one of the following schematic depicts the potential relationship among subunits 2O, 2A, 2B of RNA polymerase 2? So 2A to 2O by kinase action and 2O to 2A by phosphatase action. Then both these will with the action of protease will cause 2B. So the first option, that is option ID 345, is correct. Then next one. Mitochondrial protein synthesis is of prokaryotic origin. Following statements are being made about the ribosomes from bacteria and mitochondria. The bacterial ribosome consists of small and large subunits of 30S and 50S respectively, whereas in the mitochondria of mammals, these subunits are 28S and 39S. That is correct. In the bacterial ribosomes, the RNA protein ratio is about 2:1, whereas in mitochondria ribosome, this ratio is usually 1:2. That is also correct. Both the bacterial and mitochondrial ribosome consist of 30S and 50S subunits. That is not correct. Both the bacterial and mitochondrial ribosome consist of RNA and protein in the ratio 1:1. No, it is 2:1 and 1:2 in bacteria and mitochondria respectively. So only A and B are correct. Then next one. DNA replication occurs in S phase. The entry of cells into S phase is regulated by the tumor suppressor protein RB, retinoblastoma. The statement below are made with reference to the role of RB. RB binds to E2F in the cytosol and prevents E2F entry into the nucleus. That is correct. RB is phosphorylated by cyclin A CDK4. That is not correct. Phospho-RB activates E2F. That is correct. E2F activates cyclin E production, which promotes the G1S transition. That is also correct. So the answer here is A, C, D only. B is not correct. Then coming to shift 2, 17 to 22, shift 2. Given below are set of enzymes in the column A and enzyme activities in column B. Choose the option that matches the contents of column A with that of column B. DNA topoisomerase 1. It is single-stranded nicking. So A4. So it comes in option ID 705 and 707. The other two can be eliminated. Then DNA topoisomerase 2. DNA topoisomerase 2 double-strand break and ligation. B3. B3 is also there in 705 and 707. Polymerase epsilon. Epsilon is involved in leading strand synthesis. That is correct. And polymerase delta is involved in synthesis of Okazaki fragments. That is also correct. So option 1 is the right answer. A4, B3, C2, D1. Then next one. Precise recognition of tRNAs by their cognate amino-acyl tRNA synthetases is crucial for the fidelity of protein synthesis. In the context of amino-acylation of tRNA, alanine with its cognate amino-acyl tRNA synthetase, alanine ALA-RS, and based on the studies on the molecules of Escherichia coli origin, following statements are made. Which one of the statement is incorrect? Anticodon of tRNA alanine makes important contribution to the specificity of its aminoacylation by alanine aminoacyl tRNA synthetase. That itself is incorrect statement. So option 1 is the right answer. Then next one. Which of the following statement is not correct feature of Escherichia coli RNA polymerase? Presence of sigma subunit along with alpha 2 beta beta dash omega core RNA polymerase is required for its promoter specific binding. That is correct. Presence of sigma subunit along with alpha 2 beta beta dash omega core polymerase is not necessary for the core RNA polymerase. That is also correct. It is not necessary. That is also correct. Then mutations in the beta subunit in its rifampicin resistance defining region confer rifampicin resistance phenotype. So rifampicin, it is used in the TB treatment, and the action of rifampicin is on the ROP-B gene encoding RNA polymerase beta subunit. It is said that more than 95% rifampicin resistant mutations are associated with mutations in the ROP-B gene. The majority of mutations in ROP-B gene are present within the 81 base pair rifampicin resistance defining region, RRDR. So mutation in the beta subunit in its rifampicin resistance defining region confers rifampicin resistance phenotype. That is correct because mutations happens in the beta subunit of RNA polymerase. Then mutation in sigma subunit, not sigma subunit, mutation in the beta subunit. So option that is correct here is they have asked which is not correct. So A, B, C are correct. The one which is not correct is option D and your answer is option D. Then next one. In the figure above, replication of DNA beginning from the origin of replication of the chromosome of a newly identified bacterium having a double-stranded circular DNA genome is shown. Characterization of DNA polymerase responsible for genome replication showed that DNA synthesis occurred in the 5' to 3' direction and it depends on the presence of a primer as in the case of E. coli. Polarities of DNA 5', 3' are as shown. Replication begins at a point marked O on the left of the bubble. Both the parent strands are replicated concurrently. The longer arrow inside the bubble shows the leading strand whereas the shorter arrow shows the Okazaki fragment. The model depicts. So here it has got only one origin of replication. If it was bidirectional, then two origin of replication would have occurred here. One over here and one over here. But that is not the case. And here will be the replication fork movement in bidirectional replication. Whereas here there is only one ORI sequence over there and the replication fork is moving like this. So it is unidirectional mode of replication. And how are the sequence in which Okazaki fragments are synthesized? So C occurs prior to those marked A and B. C is the first one to be synthesized followed by A and B. So we will see the options in the next slide. Bidirectional mode of replication, that itself is not correct. Bidirectional mode of replication again in option B, that is also not correct. Unidirectional mode of replication wherein synthesis of the Okazaki fragment marked C occurs prior to those marked A and B, that is correct. Unidirectional mode of replication wherein synthesis of Okazaki fragment marked A occurs prior, not A, it is the C one that is occurring first, then only A and B. So the answer here is option 3. So coming to the year September 22, shift 1. First question. Which one of the following statements about the recognition of tRNAs by their cognate amino-acyl tRNA synthetase is correct? Amino-acyl tRNA synthetase recognize their cognate tRNAs by the exclusive recognition of their, not only exclusive recognition of anticodons. So that is not correct. Amino-acyl tRNA synthetases recognize their cognate tRNAs by recognition of their anticodons in some tRNAs only. That is correct. So they have asked for the correct statement and option 2 is the correct answer. Then next one. Which of the following options correctly matches the proteins involved in transcription, that is column A, with the DNA binding domains they carry, column B? So TF3A, it has zinc finger motif. A2, Myo-D is having helix loop helix, JUN is having helix turn helix and CRO is, JUN is leucine zipper and CRO is helix turn helix. So the option here which is correct is option ID 44. Fourth option. A2, B3, C4, D1. Then next one. The initiation of transcription is a complex process involving promoter recognition, conversion of the initiation complex from closed to open form, abortive initiation events and finally promoter escape. The following statements are made regarding these steps in transcription initiation. Which of the following option represents the combination of all correct statements? They have asked for all correct statements. Promoter escape in bacteria is usually accompanied by the release of sigma factor from the RNA polymerase holoenzyme complex. That is correct. Abortive initiation events in prokaryotes result in the formation of a short transcript approximately 10 nucleotides in length, while such events in eukaryotes result in formation of transcripts approximately 75 nucleotides in length. There is no abortive initiation event in prokaryotes, so B is not correct. Promoter escape in eukaryotes is accompanied by the phosphorylation of the RNA polymerase large subunit on its C-terminal domain. That is correct. Promoter escape. That is correct. Promoter recognition in bacteria is governed by sigma factor, which binds to the -10, -35 regions of the promoter, followed by recruitment of the RNA polymerase II core enzyme to form the holoenzyme. It is first RNA polymerase II that binds. That is the core enzyme first, and then only the sigma factor binds. So D is also not correct. So the answer here is A and C only. Option ID 257. Then next one. Although introns are not a part of the processed transcript that gets translated, they are important for several reasons. The following statements are made with reference to the possible ways in which introns are crucial to cell survival. Which one of the following options represent the combination of all correct statements? They permit the generation of different protein products from the same gene. That is correct. They may encode miRNAs which modulate the expression of gene. That is also correct. They often encode peptides which play a role in the regulating gene expression. They do not encode any peptides. So C is not correct. They promote export of certain mRNAs through the recruitment of transport proteins by the exon junction complex. That is correct. Then, they play a role in mRNA surveillance through the modulation of nonsense-mediated RNA decay via the exon junction complex. That is also correct. So, A, B, D, E are correct. C is the only incorrect statement, and your option is option ID 255. Then next one. A nonsense mutation in the protein coding region of an upstream gene of a group of genes in an operon often lead to depletion of the downstream gene products. This is a classical example of the phenomenon of polar effect of the mutation. Following statements are made about this phenomenon. Choose the option that represent all correct statements. So first statement, it occurs primarily because the termination codon generated in the upstream open reading frame lead to termination of protein synthesis, depleting the ribosomes for translation of the downstream ORFs, but it does not affect the process of transcription. It does affect the process of transcription. So option A is not correct. The phenomenon of polar effect of mutation occurs only in operons where the point mutation leading to creation of nonsense mutation also leads to formation of a stem loop structure resulting in row independent termination. It is not row independent termination, it is row dependent termination. So B is also not correct. While the presence of termination codon in the upstream ORF may deplete the ribosome that travel down to the downstream ORF, the depletion of ribosomes downstream of the nonsense mutation allows loading of the row factor that then results in premature transcription termination. That is correct. Presence of the suppressor tRNA reading the nonsense codon generated by the mutation is essential for causing a polar effect of the mutation. No. Presence of the suppressor tRNA reading the nonsense codon generated by the mutation diminishes the consequence of the polar effect. That is correct. So C and E are the only options that are correct. And your answer is option ID 267, C and E. Then next one. Regulation of mRNA translation is a major mechanism that maintains stoichiometric availability of ribosomal proteins, R proteins, to the rRNA molecule they bind to. Translation regulation is facilitated by general occurrence of the R protein genes in several operons containing multiple genes. Which one of the following represents an established mechanism to ensure optimal production of the R proteins in E. coli when the R proteins accumulate in free form, that is molar excess over rRNA? The free proteins bind to corresponding DNA sequence and activate transcription of RNA genes to increase rRNA availability. They have asked about activation. Inhibition is, they have asked about inhibition, not activation. So the first option is not correct. The free R proteins bind to the RNA polymerase. They do not bind to RNA polymerase. The free R protein bind to mRNA. That is correct. And downregulate their translation. That is correct. So option 3 is the correct answer. The free R protein bind free NTPs. They don't bind to free NTPs. They bind to mRNA. Then next one. Fidelity of protein synthesis depends to a large extent on the accuracy of amino acylation of tRNAs with correct amino acid. However, given that the side chain of many amino acids are not sufficiently different, amino acyl tRNA synthetases are often prone to misacylate the tRNAs. One such example of misacylation is of tRNA threonine by threonine amino acyl tRNA synthetase. In this context, following statements are being made about E. coli THR-RS. It misacylates tRNA threonine equally with serine and cysteine. Serine, okay, there is some structural similarity, but threonine and cysteine are not structurally similar. So first option is not correct. They have asked again for all correct statements. It possesses a distinct editing site that preferentially deacylates the misacylated tRNA threonine. That is correct. The editing of misacylated tRNA threonine occurs frequently in the cis before the release of the misacylated tRNA threonine. That is also correct. It possesses a distinct editing site that does not discriminate. It discriminates. It does discriminate between the misacylated tRNA and tyrosine tRNA threonine. Threonine tRNA threonine. The amino acylation and the editing sites of the threonine RS are the same. No. So E is also not correct. So B and C are the correct options and your option ID is 262. Then next one. Excision repair systems replace a short stretch of DNA around the site of damage. The following statements are made about nucleotide excision repair in E. coli. Combination of all correct statements. UVRB homodimer creates the nick on one strand on both sides of the lesion. UVRB recruits UVRC and UVRC creates the nick. So first option, that is A, is not correct. The 50-60 residue long stretch of DNA between the two nicks is removed by the action of UVRD. It is 12 to 13 nucleotide is removed, not 50 to 60. Then next one. The gap generated is filled in typically by DNA polymerase 1. That is correct. The distortion caused by the lesion is recognized and bound by UVRA-UVRB complex. That is also correct. So C and D are the only options which are correct. And your answer is option ID 251, C and D only. Then next one. In eukaryotes, DNA replication must occur with extreme accuracy and only once to prevent the damaging effects of gene amplification. The following statements were made regarding possible mechanisms involved in achieving tight regulation of DNA replication. High APC-C activity in mitosis and early G1 phase of the cell cycle that triggers the destruction of CDT1 inhibitor geminine, thus allowing CDT1 to be active in the early G1 to load helicases. That is correct. APC is anaphase promoting complex. Then B. Activation of SCDKs that regulate the phosphorylation of specific initiator proteins in the S phase. That is also correct. MCM helicase loads at the S phase of the cell cycle. No, it loads during the early G1 phase. Then next one. D. CDC6-CDT1 bind to the origin of replication complex and help in the pre-replicative complex assembly only after mitosis. That is also correct. So A, B, D are correct. C is not the correct option. So A, B, D only is the right answer. And your option ID is 243. Then next one. Following statements were made about chromatin remodeling in eukaryotes. Chromatin remodeling completely alters and or slides the nucleosome but cannot displace it. It can displace it. So first option is incorrect. Here also they have asked for combination of correct statements. Chromatin remodeling is an energy-driven, developmentally regulated active process. That is correct. Histone acetylation is a reversible process in which each direction of the reaction is catalyzed by different enzymes. That is correct. Then next one. In general, acetylation of the core histone reduces their affinity for DNA and destabilizes the chromatin structure, causing transcriptional repression. It is not transcriptional repression, it is transcriptional expression. So D is incorrect. Phosphorylation of serine 1 of histone H2A has been associated with transcription repression. That is correct. So B, C, E are correct and your option ID is 231. Coming to year September 22, shift to question paper. In Saccharomyces cerevisiae, DNA replication is tightly controlled and DNA should replicate once per cell cycle. Choose the incorrect statement regarding why cells do not replicate their DNA in the S phase. Pre-replicative complex remains bound to the DNA in S phase and does not allow the re-replication. That is itself is incorrect. So your answer is option 1. The next one. The post-translational modifications in one or more core histones that are known to be associated with the DNA repair pathways are. So it is ubiquitination of specific lysine residues. Option ID 702. Then next one. The long DNA strand depicted below is serving as template for lagging strand DNA synthesis. Short lines represent the newly synthesized Okazaki fragments. A, B, A, then this line, they represent the Okazaki fragments. At which position among A, B, C, D would DNA primase act next? That is at D. Sorry. DNA primase catalyze the synthesis of short RNA molecules used as primers for DNA polymerase. So option D is the right answer. Option ID 700. Next one. Which one of the following statements about DNA replication is incorrect? Once DNA replication commences, it always continues uninterrupted until the entire process is complete. Interruptions takes place. So it is incorrect. So that statement itself is incorrect. So the correct answer is option ID 693. Then next one. The following statement refer to the E. coli replicative DNA polymerase. DNA polymerase 1 displays very limited processivity and possesses 3' 5' exonuclease activity, allowing fidelity of DNA replication. A is correct. They have asked for all correct statements. So A is correct. Then DNA polymerase 3 is suitable for leading strand DNA synthesis due to its high processivity and 5' to 3' exonuclease activity that removes incorrect nucleotides incorporated during DNA synthesis. So high processivity and 5' 3' exonuclease. DNA polymerase 3 is having 5' 3' polymerase activity. So statement B is incorrect. DNA polymerase 1 possesses 5' 3' exonuclease activity which allows removal of the RNA primer, while its 5' 3' polymerase activity allows it to fill the gap created by removal of the RNA primer. That is correct. DNA polymerase 3 is suitable for lagging strand DNA synthesis due to its low processivity and 5' 3' exonuclease activity. It is having 5' 3' polymerase activity. So option D is also not correct. So the correct statements are A and C, and that is option 4, option ID 908. Then next one. Some steps in the process of eukaryotic DNA replication mentioned below require hydrolysis of ATP. Choose the following option that completely identifies all the steps utilizing ATP hydrolysis. Phosphodiester bond formation? No. DNA strand separation by helicase? Yes. Clamp loader association with clamp and DNA? No. Joining of Okazaki fragments? Yes. So the answer here is B and D only. Answer is option 4, that is option ID 920.